Hi, everybody. Welcome to the Needham Healthcare Conference. We are pleased to introduce the President and CEO of Sutro Biopharma, Bill Newell.
Thank you very much, and thank you to everybody for sticking it out here at the end of the day. It's my pleasure to present Sutro Biopharma. As we are now a public company, we're going to be making some forward-looking statements, and this slide is intended to tell you everything you should know to consider those forward-looking statements in the context of an investment decision. A year ago, we were not a public company, and a year ago, we had not started our first clinical trial. Today, we have two ongoing clinical trials in cancer. The first trial is of STRO-001, which is our anti-CD74 antibody-drug conjugate targeting myeloma and non-Hodgkin's lymphoma. That trial started in April of last year, late April. The second trial is our STRO-002 molecule. It is an anti-folate receptor alpha antibody-drug conjugate that is targeting ovarian cancer and endometrial cancer. So today we have two drugs in the clinic coming from our very novel cell-free protein synthesis platform, which I'm going to talk with you a little bit about today as well. We, in fact, are the only company in the world that has a scalable cell-free manufacturing platform technology that also encompasses a manufacturing facility that has produced not one, not two, but three drugs under cGMP conditions. Quite unusual for a small biotech and a really important and strategic asset for our company. We have the best partners. We have Merck partnership I'll tell you a little bit about that we entered into last year. We have Celgene, a very important partner of ours. We have EMD Serono as well. These partners have given us over $350 million to move our platform forward and to engage with them in multiple targets, and I'll talk a little bit about the construct of those relationships as we move forward. We have an outstanding senior leadership team with over 200 years of industry experience, and they are really the backbone that gets the job done, and that's what we do. We get stuff done. When we tell you we're going to put a drug in the clinic, we drive hard to meet the timelines to put those drugs in the clinic. We now have two, and we are on the cusp of a third IND, and I'll talk about that in a moment. What have we done most recently? Well, in the last quarter, we closed our IPO. This was an upsized round from $75 to $85 million. As a result of the Merck deal that we concluded earlier that year, they co-invested $10 million in a private placement alongside. We thought it was important for us to seek orphan drug designation for STRO-001 and multiple myeloma, and on the strength of preclinical data, which we thought might be a stretch, but on the strength of preclinical data, we were able to get that orphan drug designation. I'll be talking about our initial presentation of data at EHA in a little bit. STRO-002, we promised that we would file that IND. We did that last year. As I said, the trial is now open, and the first patient was recruited and enrolled in March of this year. And we also achieved a $10 million milestone from Celgene. It's an important manufacturing milestone. If I have time, I'll explain a little bit about it, but it really bolsters our manufacturing platform as we look towards larger scale production of the molecules that we make using our cell-free protein synthesis technology. Importantly, we ended the year with over $204 million in cash, which provides us with a cash runway into 2021. It's a very healthy runway, and what I want you to do is think about some of the next slides that I'm going to present and the news flow and the data sets that we're going to have emerge, and understand that that cash runway gives us lots of opportunities to do impactful clinical data as we move forward into 2019 and 2020. This is the slide that everybody, I think, really wants to know about. What are you going to do for us this year? What are you going to do for us in the clinic? We've been around a while. The company was formed in 2003. We've gotten two molecules into man, and now we have some opportunities to share data. For STRO-001, our anti-CD74 ADC, that trial, as I said, started in April of last year. We expect to present data at EHA in June, and that will be our initial safety data. Of course, you don't only present safety data when you're at a conference like this. You present all the data that's available to you in the study, and that's what we intend to do. So in addition to safety, to the extent that we have efficacy data, we will talk about that efficacy data. To the extent we have PK data, we will talk about that PK data. This is a little bit less than a year's worth of data as of the time we submitted the abstract. We will be updating the abstract when we make our presentation in June. Beyond that, however, we will be presenting a more robust data set, more mature safety, but also importantly, a more mature efficacy data set at the end of the year. And I'm expecting that may likely be at ASH. So there are two opportunities to understand the importance of our platform in the treatment of myeloma and non-Hodgkin's lymphoma. Ordinarily, most people don't get too excited about safety data because you anticipate that safety data is going to be good enough to keep the trial moving forward. In the antibody-drug conjugate field, however, many drugs have failed for safety. And so starting to understand the profile and the therapeutic window of the molecule by seeing the initial safety data for our first program under our platform, we think is going to be a very important tell sign as to what the opportunity is for our STRO-1 molecule. In addition, STRO-2, as I indicated, started clinical development in March of this year, and based on the clinical trial design, and I'll talk about both of these in more detail, we expect that we'll have initial safety data for that by the end of the year as well. So there are two assets that we have, one for hematological malignancies, the other for solid tumors, where the platform that I'm about to talk to you about gets to be showcased and gets to show proof of principle in the design elements that we're bringing to bear on these really important cancer therapies. Our third program is a BCMA ADC. This is a collaboration program with Celgene. It's a very high priority program for them. To give you a sense of the urgency here, we started this program from a concept that Celgene shared with us in 2016, January of 2016, and they are on the cusp of IND submission and trial initiation. When that trial is open, we will have 3 clinical development programs moving forward aggressively to treat cancer. And there's a whole wealth of programs behind those. So this slide really captures where we are today. STRO-1 and STRO-2 I've already talked about a little bit. They are wholly owned by Sutro. We have multiple oncology and immuno-oncology programs behind them, and I look forward to declaring what STRO-3 is going to be. I expect to be talking about STRO-3 at the end of the year when we have a sight line on when we would be filing an IND for that program. There are several that we are competing right now, and we want to take the best one forward to be our third wholly owned clinical program. I mentioned the Merck transaction, and we have an exciting relationship with Merck that started in July of last year in the oncology and autoimmune space. Celgene has 4 programs, not just the BCMA ADC, but 3 other bispecific antibodies that we're excited about and look forward to their advancement ultimately into the clinic. And finally, we have a relationship with EMD Serono. It's a multi-target relationship, and the lead program is a bispecific drug conjugate. So there's a lot of stuff going on here in what I call the ABCs of next-generation cancer therapy: antibody-drug conjugates, bispecific antibodies, and cytokine derivatives. There are no company, certainly not a company of our size in the world, that is able to have that many different modalities and its own GMP manufacturing facility. That has been built in this company over the last 16 years. Let me talk a little bit about our collaborators, Merck, Celgene, and some of the others. As I said, we were pleased to be able to announce the deal with Merck in mid-2018. It is in the cytokine derivative space, a very important space for those of you who might be familiar with Nektar-214 in combination with checkpoint inhibitors. Bristol saw a tremendous opportunity to amp up the profile of their molecule, and they locked up Nektar-214. Nektar-214 is a very complex molecule. It's an IL-2 cytokine that has 6 sites of pegylation, which have to be slowly revealed in order for the drug to have the efficacy potential and not have out-of-control toxicity associated with it. We apply very rational structure-based design principles that I'll tell you about in a minute. And when we think about a molecule like that, we wonder whether it is the optimal molecule. Yes, it is good enough and has shown a lot of promise in combination therapy, but is it the optimal molecule, or could you do better? Merck's bet with us is that we could do better. This is the second largest preclinical immuno-oncology deal that Merck has ever done, and is really a testament to their faith and our ability to rapidly work with them in collaboration to develop a cytokine derivative that is powerful and impactful for their oncology and their autoimmune franchise. We received $90 million in funding from them in 2018, and we have substantial over $1.5 billion in milestones and royalties in the mid-single digit to low double digit. This is an unusual deal in the sense that we did not come to Merck and say, we have an asset like Nectar-214, we'd like deal terms like this for the asset. This was a concept where we were in the cytokine space and we said, let's merge the knowledge that we have with the knowledge that you have. Let's use our really outstanding protein engineering skills, and let's drive to not find a molecule that's good enough, let's actually find one that's best in class. And that's what we do with all of our programs. Celgene is a different relationship. We've been involved with Celgene since 2012. Right now, as we sit here today, we have narrowed our relationship down to 4 programs. The BCMA ADC, which when it goes in the clinic, they will own worldwide rights to. 3 other bispecific antibodies. Of those other 3 bispecific antibodies, assuming they make certain payments to us, they have the ability to own the worldwide rights to the 2nd program to go in the clinic. The 3rd and 4th programs we will own U.S. rights to. They will not have the ability to acquire those U.S. rights. So as these programs advance, we actually have hidden additional programs in the sense that these collaborative efforts with Celgene can yield 2 additional programs that we own U.S. clinical development rights to. They've been very generous in their funding, $220 million to date from them, and we've got very significant milestones and royalties. EMD Serono is an earlier deal from 2014. I think the novelty of the molecule, when we can talk about it, a bispecific drug conjugate, will speak for itself. That's one that we've been moving forward with them, and I'm excited about and hope to talk about that in more detail later this year, early next. Not to be forgotten, our platform technology just doesn't cover the ABCs of cancer, antibody drug conjugates, bispecific antibodies, or cytokine derivatives. Our platform technology also enables really complex vaccines. And we spun out Sutrovax in order to take advantage of that platform technology. They're working on a 24-valent pneumococcal conjugate vaccine. That would be far more protective than the existing vaccines, whether it's Prevnar 13 or Pneumovax or Prevnar and Pneumovax in concert. They've successfully raised $170 million in equity funding, and they're still at the preclinical stage. They are looking forward to starting clinical development in the next several years. Importantly for us, even today, we still have a 5.6% ownership percentage of this company, and we have a single-digit royalties. I am on the board, and so I get to see the exciting progress that Sutrovax is making and using our technology in an area that, frankly, we wouldn't have been able to afford to do and didn't have the focus to do. We're going to be focused on cancer. They're focusing on vaccines. So let me talk to you a little bit about how we got here. When I joined the company in 2009, we had a novel technology that had been spun out from Stanford in 2003. It's called cell-free protein synthesis. Let me deconstruct that for you to begin with. Virtually every other complex large molecule is made in CHO or E. coli. The cell is the factory. Our founder, Jim Swartz, a professor at Stanford, ex-Genentech, thought, in vitro transcription and translation has been around for a long time. Simply put, we can take the inner workings of the cell, the ribosomal machinery and other associated machinery, and pull it outside of a cell and put it in a test tube. And we can keep that machinery active. Now, when it comes time to do the protein synthesis, you take DNA, you insert it into the vessel where that lysate is, that ribosome-rich lysate is, and as the machinery does its job, a fully formed protein is formed in 8 to 10 hours from DNA. If we want to have larger yields, we run the reaction for 12 to 15 hours. But in less than a day, we can make a complex large molecule. We don't rely on the cell growth, we rely on the volume of the vessel to give us the yields at gram per liter yields. What does that allow us to do? What it means is when you're looking at very complicated molecules, an antibody-drug conjugate, an ADC, is an antibody with a linker and a cytotoxic warhead attached to it. How that molecule gets made is very difficult. If you have to rely on cell lines in order to make your molecule, every cell line is different for every different molecule that you wish to try. So after you've tried 4, 5, 6, 7, you've pretty much exhausted a lot of resources, particularly if you've gone up to scaling it so that you can do larger animal studies. In a cell-free protein synthesis environment, quite simply, we just make many variants overnight, and then we start to triage them, understanding which ones have the exact properties that we're looking for. And so we're able to select the optimal candidates, not by settling for something that's good enough, but by actually saying, does this have all the properties I want? Is it rapidly internalized? Is the warhead released in the right place? What is the molecule that is formed and that gets released that creates toxicity, not just to the tumor cell, but to the rest of the healthy cells? We're able to tease that apart and then play with the molecules rapidly to figure out what is the best one that has the right properties and, most importantly, the right therapeutic window. If you're going to be dosing a cytotoxic, you want to be able to be efficient in your dosing, and you want to have the right therapeutic window so that the patient doesn't have to go off drug because of unnecessary toxicity. We think first-generation ADCs and many of the other molecules that are in the clinic, they are just good enough, but they have unnecessary toxicity, and that limits them from creating their full potential. Because we do not have to create a stable cell line to move our program forward, what we work on at the bench is the same fundamental system that we use at larger scale in our GMP manufacturing facility. Because we don't have to do that stable cell line, we can accelerate the time to IND by about a year. And that's remarkable, particularly when you're in a very competitive field. Our first program, STRO-001, is a first-in-class molecule, but STRO-002 is going to be a best-in-class molecule, and so being able to move to the clinic faster not only saves money, but it gets the drug to the patients much more quickly. And this is a platform that generates homogeneous assets, whether it's ADCs, bispecifics, or cytokine-based therapeutics. We think homogeneity is the only way to tease apart the relevant pharmacology and the toxicity to optimize the value of the molecule. For us, we try and pattern ourselves after other companies. You can think about a lot of leading companies that have rapid protein discovery. I would say Ablynx or MorphoSys are probably two of the more significant names who have that. You can think about companies that have precise protein engineering. I would say Xencor and Macrogenics claim to have very precise protein engineering. But no one is able to marry those two functionalities together in the way that Sutro is able to, and no one has a commercial scale manufacturing capability that we control that has the ability to spray dry the extract, the starting material that is the basis for all the molecules we make. It's said that, one cell line is one drug, but for us, our extract produces all of the drugs in that pipeline slide that I showed you, and that gives us a tremendous amount of efficiency and flexibility. So that's a little bit on the platform. Let me talk a bit now about STRO-001, our anti-CD74 ADC. Why are we in B-cell malignancies? Well, the fact of the matter is, for most B-cell malignancies, I'll leave DLBCL to the side for a minute, patients are not cured. There are tremendous therapeutic improvements that have arisen over the last several years, but nonetheless, we see breakthroughs. In our clinical trial, we even see breakthroughs from CAR-Ts already. So patients need to get the next therapy. A therapy that has a good therapeutic window, that is well tolerated by the patient, but is able to delay the progression of their cancer, that would be a great therapy. And as a first-in-class therapy, we then succeed to a market that has a prevalence that's over 600 million today. So that's a large market, and all of those patients are going to blow through even the most exciting novel therapies, and they're going to need something else. CD74 is our answer to what they need. Now, When we thought about CD74, we thought, well, this is a great target. It's expressed on B cells. It's minimally expressed on other cells. That's a great thing if you're going to be targeting a cytotoxic. And importantly, the molecule is rapidly internalized. So let me talk to you a little bit about how we thought about designing the molecule using the toolkit that I've just talked about. In the antibody-drug conjugate field, many people say that the drug-antibody ratio, how many warheads are stuck to any individual antibody, should be 4. And there's some reasons using the first-generation technologies that they would conclude that. 2 didn't give you enough potency, and more than 4 made the molecule a mess, and it really wasn't able to be delivered to the patient, and so 4 must be the magic number. Well, in fact, if you're making a heterogeneous mixture, that is what you might well conclude. But when you can make a single species that is optimized, you might find when you're thinking about therapeutic window that you get a really good killing effect from a drug-antibody ratio of 2, and you don't have the excess toxicity that a drug-antibody ratio of 4 or 6 would give you. And in fact, that's what we found. So we have a stable, non-cleavable linker with a DAR of 2 for this molecule. The benefit of the stable linker is that when the tumor cell takes the antibody-drug conjugate in, what is the killing moiety is the non-natural amino acid that is proprietary to us that we use to be the site of linkage for every antibody-drug conjugate we want to make. It's that non-natural amino acid that's a linker and it's a warhead. And that whole thing is very toxic inside the cell, but it is so large when it's released as the cell dies, it is hydrophilic and does not penetrate other cells easily. As a consequence, we're minimizing the bystander effect. We spent a lot of time looking at where this warhead should be placed to identify the best spot in all the antibody to place our linker and warhead. I've already talked about the hydrophilic nature of the catabolite. That's what I was talking about before. And really our homogeneous design gives us this single product species, which we think means we're going to have something that is more efficient at targeting the drug conjugate, the payload, to the tumor microenvironment. Now, I look forward to talking at EHA about human data, but until we present that, you're going to have to bear with me on our animal data, because this is frankly quite exciting and another reason why we went into the clinic with this molecule. So we looked at STRO-1 in animal models of multiple myeloma, and here is a bioluminescence imaging of tumor cells. You take a healthy animal and you inject tumor cells into them, myeloma cells, and you see on day 7 both the vehicle-treated animal and an animal that was being treated with a single dose, 1 mg per kg of our STRO-1, their tumor uptake. Over the course of 28 days, the animal that is treated with vehicle ends up lighting up with tumor cells. That's a very rapid progression. Yet at 1 mg per kg, and also true at 3 and 10, but at the lowest dose 1, day 28, you actually see no tumor cells in the imaging. When you look at how these animals did for survival, the ones that were dosed at 10 mg per kg, 5 of those 6 animals on a single dose, not the way you would treat a patient, but on a single dose, 5 of those 6 animals survived. And you see a dose-dependent improvement in survival as you go from 1 to 3 to 10 mg per kg. That's pretty impressive in terms of survival for a single dose. We also looked at lymphomas, and there are models for diffuse large B-cell lymphoma and mantle cell lymphoma, and we thought these were very exciting as well. So in this DLBCL model, what you see is that animals that are treated with vehicle, once the tumor is established, it grows and grows quite rapidly. And then you see a dose-dependent effect when you go from 1 to 3 to 10 mg per kg. That tumor growth slows down at 1 and 3 mg per kg, and even at 10 mg per kg, a single IV dose, out to day 90, all of those animals show no evidence of tumor cells. So that's a really powerful model of diffuse large B-cell lymphoma. In a mantle cell model, a murine xenograft model, we did the same thing, looked at different doses of our STRO-1, and what you see is survival out to day 135 at the highest dose level for a single dose, and at 3 mg per kg, the vast majority of the animals are also surviving. Now, the animal studies are great. These were important validation for us to move our molecule into the clinic, and we're waiting for the clinical trial results to be able to establish that in man our molecule performs as well as possible. Now, our trial design is a little intriguing in the sense that it's not one study, but it's really two studies in one. And by that I mean we have two dosing cohorts. One is for myeloma patients, and the other is for lymphoma patients. Why do we have that? Well, the diseases, even though they're both B-cell malignancies, have different natural histories. They're really different in the way they present, and they have different courses of therapy. And so we want to understand, how does our drug work in both of these? And the way to do that is to have two separate cohorts. We started at the FDA recommended dose level for both of them, and over time we move up dose escalation. So in the early days, because we were starting at relatively low, what we believe to be subtherapeutic doses, we did single patient cohorts in order to get to the next higher dose as quickly as possible. And when we achieve a safety signal for both arms, the study automatically converts into what is traditionally known as a 3+3 design, and that gives you more patients at various dose levels. And so as we move into what we think are the therapeutically relevant doses, you will see 3+3 data. Once we reach our maximum tolerated dose, we will then be able to determine our recommended phase 2 dose. They may be the same or they may be different depending on what we see in dose escalation. And then we have an option for 4 different dose expansion cohorts: myeloma, diffuse large B-cell lymphoma, mantle cell, and follicular lymphoma. We will use the data from our dose escalation to help define which cohorts we want to go into. Maybe we'll go into some first and some later. Maybe we'll go into all at the same time. It will really depend on the data. But we have an opportunity in the trial design to really understand, when this study is fully enrolled, what is the potential of our drug to treat this very serious disease? And that's an exciting transformation for our company, and we have the capital to move this study forward. The second molecule, STRO-002, is a folate receptor alpha molecule. We are excited about the opportunity to have a best-in-class molecule here to treat both ovarian and endometrial cancers. A lot of times people wonder, is this a validated target? I think the consensus from a competitor molecule that just did not succeed to achieve statistical significance, the ImmunoGen molecule, mirvetuximab, the consensus is it proved that the target is a validated target, particularly in the high expressors of folate receptor alpha. And so this is a really exciting program for us because, frankly, they are not on the same path, because their study failed, that they were on even a month ago. That opportunity is one we are looking forward to really taking advantage of. And why are we looking forward to taking advantage of it? We have a completely different molecule. Again, we apply the same design principles, but because this is a solid tumor, we come up with a very different result. In this case, we still want a homogeneous antibody-drug conjugate. That's very important because it allows us to compete out versions to find the most efficacious version. In this case, we wanted a drug-antibody ratio of 4 because we found that 2 wasn't sufficiently potent in a solid tumor environment. 4 was the optimal one when we think about efficient killing of the tumor and having a proper therapeutic window. In this case, we started with a different warhead. This is still a tubulin inhibitor. Our first molecule had a maytansinoid derivative. This is a hemiasterlin derivative. This is an important warhead because it has some wonderful properties, not only in killing the tumor cells, but in the way the body excretes it. In this case, we have a cleavable linker, not a non-cleavable linker. And why is that? Well, when the warhead gets released, this hemiasterlin has a half-life of only 2.5 hours. So inside a tumor, it's very effective. When it gets released, that 2.5-hour half-life means it's cleared rapidly. The mirvetuximab molecule has a maytansinoid warhead that has a 25 to 40-hour half-life. So when it gets released, after it's done its job, it's also creating collateral damage that may be dose-limiting in terms of the toxicity and may have limited the therapeutic utility of their molecule. So we looked at this molecule in models of ovarian cancer, and this OVCAR-3 murine ovarian cancer model is a very difficult one to do. But we grew the tumor cells up and treated with vehicle. The animals don't do well. But a single dose of 5 mg per kg of STRO-002 controls the tumor for a while. More importantly, a single dose at 10 mg per kg has a better effect in terms of knocking the tumor down and maintaining that control longer. This was an exciting animal model for us, and one of the reasons that we decided to move this program forward into the clinic. Another reason we decided to move this forward into the clinic, and frankly how we got excited about this program, was that we thought, heterogeneous ADCs like mirvetuximab, we should be able to prove preclinically that we can do something better than they can do. And so what we did, even before we got to the animal model, was we said, let's make a copy of mirvetuximab. We can't make it exactly the way they do because we don't have their cell line, but we do know what the antibody is, and we can make that using CHO cells. We do know what their linker warhead is. We do know how they conjugate, and we can make a molecule, and we can profile that molecule against everything they've published to see if we've been able to mimic that molecule. It's not perfect, but it's a really good substitute and one that's used a lot in the industry. And so this benchmark molecule, this heterogeneous benchmark, let's just think of that as something that's pretty darn close to the drug that failed. When you take this benchmark molecule and you put it in a cell line or in cells that are positive for the target, you see a cell killing curve, which is a respectable cell killing curve. But when you put it in cells that do not express the target and you shouldn't see any cell killing, well, lo and behold, it kills too. This blue line is the benchmark. Now, why is that? Well, this is a multi-day experiment, and what we hypothesize is happening is that the warhead is coming off and that it's killing the cells non-specifically. So it's really not a targeted therapy. It's much more like a chemotherapy, and the cell killing rate for these two curves is pretty much the same. So we thought, okay, let's see if we can do better. Well, first of all, we were able to design a molecule that had a very stable linker, and so in these folate receptor alpha negative cells, it doesn't kill at all. Okay, we've solved the linker problem. That's a good thing. And then when we have the best molecule, not a heterogeneous mixture where the four warheads on average are placed all over the molecule as they are with mirvetuximab, but one where they are precisely positioned. What happens with that molecule in the folate receptor alpha positive cells? It's 50-fold more potent. That's huge in terms of our ability to engage in cell killing, and we think that means we have opportunities for this molecule not only in the folate receptor high group, but also in the medium and potentially lower density patient population as well. So this was exciting data for us. One of the other things we looked at was what happens to that warhead after it's released. So for our molecule, we wanted something that wasn't going to slosh around, and what you see is, and here we're measuring free warhead in the tumor versus warhead in the blood after dosing. So on day one, we can detect, this is our show 2 over here, we can detect a lot of drug in the tumor. And by day 3, it's even more, and day 7, it continues to remain in the tumor, doing what it should, killing the tumor cell. But you see that in the plasma levels, it is non-detectable as well. And that is explained by the rapid clearance, we think, of our molecule. What happens with the homogeneous benchmark free warhead? Well, it accumulates a little bit on day 1, and then seems to stabilize by day 3 or day 7. But then what you see is that for them, they have plasma levels on day 1 that are sky high. They do decrease over time, but this creates toxicity that we think explains some of the dose-limiting characteristics of many ADCs. So this trial started. It's a similar design, but it's got a different approach to it. We start again with the FDA recommended dose, and we go through a dose escalation process. The first 3 cohorts in this study are single patient cohorts. And at the 4th cohort, because of the level that we expect to be at, the study automatically converts into a 3+3 design. So we started this study in the middle of March. After 3 weeks, we look at the patient and we have a safety evaluation team meeting. And if the safety evaluation team says go, we go to the second patient and they start the second dose escalation cohort. 3 weeks again, same thing up, and then we move to 3+3. So what we think is going to happen is we will get to a maximum tolerated dose. We will then have our recommended phase 2 dose, And then we have the opportunity for dose escalation in 2 cohorts, ovarian cancer patients and endometrial cancer patients. Because there is less known about endometrial cancer and molecules like ours, we will want to make certain that endometrial cancer patients do have some expression of folate receptor alpha to be enrolled in this part of the study. For the ovarian cancer patients, I think the jury is out. We're taking all comers right now, and so we want to understand how our drug profiles in low, medium, and high patients, and that'll give us some opportunities to decide where we want to go in ovarian cancer. After that, we certainly have combination therapy opportunities. This is the management team. As I said, over 200 years of experience. Our Chief Medical Officer, Artur Molina, was responsible for the development of Zytiga and Rituxan. Our Chief Scientific Officer, Trevor Hallam, has over 30 years of experience in the industry. He's really the architect of the ABCs of next-generation cancer: antibody-drug conjugates, bispecific antibodies, and cytokine derivatives. This is an exciting time for us. 2019 is going to have a lot of clinical data readouts, and I want to thank you all for your attention, and I hope this is something you will find of interest. Thanks very much.
Any questions from the crowd? Can you sort of outline use of cash in terms of where that gets you with each of these programs?
Yeah, so capital runs, present capital runs into 2021. What I expect is going to happen is that in 2020, we will see dose escalation occurring in STRO-1, and that could be starting in the early part of the year, and in the mid to latter part of the year, we may have a potential for dose escalation in STRO-2. So again, depending on how fast dose expansion in phase 2, in STRO-2. So depending on how fast we move along relative to our cash, we'll see how long that takes us. But as I said, we expect a lot of exciting data this year and really throughout 2020 as well from these programs. I should also mention the BCMA program is completely funded by Celgene. And so as that trial moves forward in 2019 and 2020, they bear the full cost of that, and they actually pay us to make the drug for them, so we make a little bit of money on that as well. Because of BMS? I don't know. We will have to talk to BMS. I think right now we felt encouraged when the announcement came out that they talked about the key myeloma franchise that Celgene brings to the combined company. In that franchise, it's not just Rev and POM. They're also looking at their next generation BCMA-targeted molecules. That's a CAR-Ts, a bispecific, and our antibody-drug conjugate. And so we were pleased to be acknowledged as one of the important components that Bristol is looking at in their acquisition of Celgene. Assuming that transpires, shareholder vote is coming up shortly, we'll have a chance to get to know Bristol better. But we're thrilled for both companies. Celgene's been a great partner, and if Bristol concludes the acquisition, we'll be excited to work with them as well. The dose escalation is in relapsed refractory, yes.
Considering there is almost no other treatment available, do you see a fast path for approval here?
A lot's going to depend. There could be. It will really depend on what the data is, and we're designing the study so that in the dose expansion phase, we can truly understand whether there is a fast path to approval for that patient population. A lot of times, if you're showing exciting therapeutic benefit and a good therapeutic window, which is, in this case, we think is important and relevant, you can either start another study or expand the study a little bit further so that on the basis of a single-arm uncontrolled study, you can foresee a path to regulatory approval. But I'd like to see much better efficacy profile than mirvetuximab showed up, and certainly a much better safety profile. Yes?
Hi, Jennifer. I have two questions about technology, and I wanted to help us understand. One is that the actual ADC linker was non-cleavable versus cleavable. So if you're designing this drug, how do you go about thinking whether or not you want this drug to be the linker to be cleavable or not? And second, you have really a great array of different types of technology platforms. When you go in like high level, when you go into the drug, how do you know which one you're going into? And I guess it's like drug development and design questions for that.
Yeah, so that's a really interesting question. The answer is there's a lot of empiricism that goes into it. Because we make molecules so quickly, I'm going to actually merge the two questions together into one. It can be the case that when we're looking at a target that we're excited about, we're wondering, is an antibody-drug conjugate the right thing to target that tumor, or do we want a bispecific antibody? And so perhaps with a CD3 killing mechanism. So in some instances, we have created both assets and then competed them empirically to decide which had the better profile that we're looking for. So whether it's cleavable or non-cleavable, we're looking at a lot of different properties preclinically to determine what's the optimal molecule. You know, the fact that the first one is not cleavable really meant that we needed to make certain that that molecule, if it was going to stay around the body, was as innocuous as possible. We don't really believe in the era of combination therapies when you're dosing cytotoxics that you really want a lot of bystander effect because you may be taking away other tools in the oncologist's armamentarium, perhaps immune regulatory tools as well. And if you've blown up all the immune cells around the tumor microenvironment, you don't have the ability to effectively combine there. So empirically, we will look at what's the nature of the molecule, what's the tumor microenvironment, what does the catabolite look like, and what warhead are we choosing? I've profiled two. Our team is looking at multiple different warheads of multiple different classes because we want to be specific based on empiricism, not say, I've got one hammer and everything must be a nail for that one hammer. I really want the whole toolbox, and that's what we've been working on these last few years. And whether you're going to go with an antibody-drug conjugate or a bispecific antibody, if you can make both and it's efficient to do so, why wouldn't you? And that's the way we approach thinking about these things.
All right, thank you.
Great. Thank you all. Appreciate it.